Welcome to the PubMed Daily Review for April 9th, 2026. Today we scanned 2,941 new open access papers. Here are the top 5 selected for detailed analysis. Trend 1 of 5, Inclusion, Resistance, and Genetic Phages. This trend explores health disparities, antimicrobial resistance, and novel genetic approaches to combat infectious diseases. This cluster covers 408 papers. Representative work includes barriers and opportunities to include underrepresented population groups in vaccine trials. Cross-sectional, observational, online survey study from the Accelerate Research Network, antimicrobial-resistant pathogens on the plate. A semi-quantitative hygiene risk evaluation of raw beef consumption in Ethiopia within a One Health context, and genetic analysis of F1 cluster phages that infect Mycobacterium smegmatis identifies 2 distinct Holon-like proteins that regulate the host lysis event. Trend 2 of 5, Digital Health and Youth Trajectories. These papers explore how digital and health interventions shape individual and group development across lifespans. This cluster covers 661 papers. Representative work includes telephone-based mental health promotion for rural women in Brazilian agrarian reform communities, pre-post pilot study, cardiac synchrony during collaborative drawing, a longitudinal comparison of same-generation and intergenerational dyads, and from instability to recovery, mapping youth housing trajectories with life history calendar. Trend 3 of 5, Cardiovascular disease and mortality trends. These papers collectively investigate cardiovascular health, disease, and mortality through diverse analytical and clinical approaches. This cluster covers 449 papers. Representative work includes concordance of death information between 2 health systems serving the same region and the Social Security Administration Death Master File. Retrospective observational study. Are serum MoC levels and MoC M.1382 A greater than C polymorphism related to polycystic ovary syndrome? And tirzepatide-induced ketoacidosis with hyperglycemia in a patient without diabetes. Trend 4 of 5, Chemical synthesis and anti-tumor agents. These papers explore chemical synthesis for diverse applications, including anti-tumor agents and material properties. This cluster covers 692 papers. Representative work includes divergent mechanisms of cranial suture ossification in normal development and pathologic fusion, highly active CH bond borylation of aromatics with pyridine as convenient, low-cost ligand under iridium catalysis, and modulating IDO1 and TDO inhibition through structural modification of diaryl hydroxylamines. Trend 5 of 5, Clinical trials and medical imaging. Advancing healthcare through clinical studies and innovative medical imaging for better diagnosis and treatment. This cluster covers 731 papers. Representative work includes progressive Achilles loading via clinician e-support, PACE protocol for a dual-site randomized controlled trial, effects of sitting Bajoangen on cancer-related fatigue in patients with advanced cancer, protocol for a randomized controlled trial, and automatic recognition and prognostic prediction of colorectal liver metastases using a multi-scale deep learning framework, model development and validation study. Up next, our first paper out of 5. Suboptimal ceftazidime-avibactam exposure drives sequential BLA, KPC, mutations, and intra-host coexistence of Klebsiella pneumoniae harboring distinct variants leading to persistent infection by Feng Yang and Fu Ping, who from Institute of Antibiotics, Huashan Hospital, Fudan University 198171 Shanghai, China. Let's break down its key contributions. KPC-producing Klebsiella pneumoniae infections are a serious and growing problem worldwide. When these infections don't respond to treatment, it's often because the bacteria develop new resistance mechanisms. We don't fully understand how these resistance mutations arise during therapy or why they can lead to infections that just won't go away. This study aimed to shed light on these critical questions. To investigate this, researchers analyzed 10 different Klebsiella pneumoniae samples collected over time from a single patient who had a persistent infection. They used a combination of advanced techniques, including whole genome sequencing, to pinpoint the genetic changes occurring in the bacteria. They also performed experiments to understand how these genetic changes affected antibiotic resistance and bacterial survival. The study found 4 different versions, or subtypes, of the BLA-KPC gene. They identified 7 distinct mutation events that occurred sequentially in the bacteria over time. These mutations progressively increased resistance to ceftazidime-avibactam, and remarkably, multiple different resistant versions of the bacteria were found living together within the same patient. This coexistence of variants was a key factor in the persistent infection. This research has important implications for how we treat difficult infections. It shows that using antibiotics at less than optimal doses can actually encourage the bacteria to become more resistant, making the infection harder to clear. The finding that multiple resistant strains can coexist within a patient underscores the need for personalized treatment approaches. Encouragingly, some newer antibiotics appear to be effective against these highly resistant bacteria, offering hope for future treatment strategies. Up next, our second paper out of 5. Engineered M2 macrophage-derived vesicles deliver DNase I for cfDNA clearance and multi-organ protection in sepsis by Fan Wu and Zhongxiu Liu from A Department of Emergency, the first affiliated hospital of Wenzhou Medical University, Wenzhou 325035, China. Let's break down its key contributions. Let's look at engineered M2 macrophage-derived vesicles deliver DNase I for cfDNA clearance and multi-organ protection in sepsis. Sepsis is a life-threatening organ dysfunction caused by a dysregulated host response to infection. Despite advances in critical care, it remains a major global health challenge, highlighting the urge. Up next, our third paper out of 5. Transfer of damaged mitochondria from cancer cells to cancer-associated fibroblasts promotes tyrosine kinase inhibitor tolerance in EGFR-mutant lung cancer by Tongyan Liu and Rong Yin from Department of Thoracic Surgery, Jiangsu Key Laboratory of Innovative Cancer Diagnosis and Therapeutics, Jiangsu Cancer Hospital and Nanjing Medical University Affiliated Cancer Hospital and Jiangsu Institute of Cancer Research, Nanjing, PR China. Let's break down its key contributions. EGFR-mutant lung cancer is often treated with tyrosine kinase inhibitors, which are highly effective initially. However, a major challenge is the development of resistance, leading to treatment failure. Emerging evidence suggests that interactions between cancer cells and their surrounding stromal cells, like cancer-associated fibroblasts, play a crucial role in this resistance. Specifically, the transfer of mitochondria between these cells via nanotubes might be a key mechanism. This study investigated a novel mechanism contributing to drug resistance in EGFR-mutant lung cancer. Researchers focused on the transfer of mitochondria between cancer cells and cancer-associated fibroblasts, which are known to support tumor growth. They explored whether blocking this nanotube-mediated mitochondrial transfer could be a viable strategy to overcome resistance to tyrosine kinase inhibitors. The core idea was to disrupt this intercellular communication to resensitize the cancer cells to treatment. The study found that pharmacologically blocking the nanotube-mediated transfer of mitochondria was indeed effective in overcoming tyrosine kinase inhibitor resistance in EGFR-mutant lung cancer. This blockade successfully enhanced the efficacy of EGFR inhibitors, suggesting that disrupting this mitochondrial exchange is a promising strategy. The results clearly demonstrated that targeting this specific intercellular mechanism can restore sensitivity to therapies that were previously failing. This research highlights a significant clinical implication. Targeting the transfer of mitochondria between cancer cells and fibroblasts presents a novel avenue to overcome drug resistance in EGFR-mutant lung cancer. By blocking this process, we can potentially resensitize tumors to existing tyrosine kinase inhibitors, offering new hope for patients who have developed resistance. This strategy could be a valuable addition to our therapeutic arsenal, and further clinical investigation is certainly warranted. Up next, our fourth paper out of 5, Uncovering Immune Niches in Health and Disease Using Spatial Transcriptomics by Johan Thorsen and Camilla Engblom from Division of Immunology and Respiratory Medicine, Department of Medicine Solna, Karolinska Institutet, and Clinical Immunology and Transfusion Medicine, Karolinska University Hospital, Stockholm, Sweden. Let's break down its key contributions. The immune system is incredibly complex, with cells organized into specialized microenvironments called immune niches. These niches are crucial for how immune cells function, whether in a healthy state, during inflammation, or in cancer. Until recently, studying these niches in their natural tissue context was challenging. Now, exciting new technologies like spatial transcriptomics are opening up unprecedented opportunities to explore these intricate cellular ecosystems. This paper is a review synthesizing current research that utilizes spatial transcriptomics. This powerful technology allows scientists to map gene expression directly within intact tissue sections, preserving the spatial relationships between cells. The focus is on how this approach has been applied to understand the cellular and molecular makeup of immune niches, particularly in intestinal and tumor tissues, offering insights into both health and disease. The review highlights that spatial transcriptomics has been instrumental in dissecting the intricate architecture of immune niches. Key applications focus on intestinal and tumor tissues, revealing insights into immune responses in health, inflammation, and cancer. Furthermore, emerging studies are now exploring the spatial organization of B and T cell clonal niches, expanding our understanding of adaptive immunity at a spatial level. The key takeaway is that spatial transcriptomics is revolutionizing our ability to study immune niches. This technology offers a powerful lens to understand complex cellular interactions in situ, which is crucial for developing new treatments. While still evolving, the future of spatial transcriptomics in immunology and oncology is incredibly bright, promising deeper insights and more targeted therapies. Up next, our fifth paper out of 5. Identification of tumor-initiating cells and early marker genes in histologically normal colonic mucosa that lead to neoplastic transformation by Sangeeta Jaiswal and Thomas D. Wong from A Division of Gastroenterology, Department of Internal Medicine, University of Michigan, Ann Arbor, Michigan, USA. Let's break down its key contributions. Colorectal cancer is a devastating disease affecting millions worldwide. We know a lot about how precancerous polyps turn into invasive cancer. But what happens even before that? How does a normal-looking colon cell first start down the path to becoming cancerous? This study aimed to uncover those earliest cellular and molecular changes. To tackle this question, the researchers used a powerful technique called single-cell RNA sequencing. They analyzed cells from both normal-looking colon tissue and matched polyps from 7 individuals. This allowed them to look at the gene expression of thousands of individual cells. They also employed other advanced methods like RNA velocity to predict cell fate and RNA fish to confirm findings in their original tissue locations. The analysis revealed a specific population of cells within the histologically normal tissue that they termed tumor-initiating cells, or TACs. These TCs were distinct from normal stem cells, showing early signs of epithelial mesenchymal transition, or EMT, and altered signaling pathways. Key marker genes like ETS2 and SLC12A2 were identified, and these findings were robustly validated using spatial techniques and an independent dataset. This research provides crucial insights into the very beginning of colorectal cancer development. By identifying these early tumor-initiating cells and their unique molecular signatures, we now have potential new targets for early detection. These findings could pave the way for novel diagnostic tools and interventions aimed at intercepting cancer development at its earliest stages. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.